really fun. And so he's really fun to be with when he has time for her, and he comes home and they, go traveling and they do things. But after a while, he's not as home very much, and you can only do so much with 10-carat diamonds. After a while, you can, play around with them in the bathtub, and then they just become every day like sand. And so she finds herself back at the bar, and this guy comes in, and my goodness, he's done so much during his life. She thought she had done a lot, but there's so many things he talks about that she finds, even though she'd never heard of them, that she's interested in. So when he invites her home, she goes right along. I told you it wasn't very flattering. And this time, the next morning, there's only one diamond in her orange juice, and it's only 5 carats. And it's not a helicopter outside, it's just a regular, jet airplane, a King. But she's been everywhere already. And he's just so much more interesting that she finds herself she doesn't have to go shopping. She finds herself enjoying just talking to him, horseback riding. And so she doesn't know that he's got just as much money as the other guy, but he's not spending it on her. He's buying CDs, and he's buying companies, and he's buying more airplanes. And she doesn't really care because it's the guy that she's enjoying so much more. She's easily bored. So in another few weeks, she's back at the bar. And she goes home with the third guy. This guy doesn't even have a mansion. And outside it's just a Honda Accord that she can drive around. And his credit card isn't even platinum. But there is money in the bank. And she spends even more time. She can't understand what it is. They don't even have to talk. They just look at each other and they understand what the other person is thinking. And she thinks that she's found the one that she wants to be with for the rest of her life, but, he gets boring too. She finds herself back at the bar. And she thought she's tried everything, but this guy comes in, he's got flip-flops on, he's got a horse tied out, and he said, he said, if you come back with me, we'll wake up to the sunset. I've got a shack on the beach. And, I've got a boat. We can go scuba diving. And she's never been scuba diving before. She hasn't been deeper than where she could snorkel. And so she finds herself on the beach with this guy. They spend every single minute together. He's not off. He's got a fortune that's why he doesn't have to work. But she doesn't use it. She doesn't need it. She's attracted to him. So hopefully, you weren't so caught up in my storytelling that you didn't miss the point that it was her attraction to the guy that brought her home with them, and they didn't have to spend as much money on her. That energy, that money that they saved, they could buy more cars and more planes and ATP molecules. So the difference between the amount of energy needed versus the chemical attraction. You have to have the chemical attraction to draw the electron away from that molecule, but because the energy level is at a lower level, that additional energy, we don't want to release it all at once. Fireworks are great, but not when you get burned up by them, okay? So we release that energy enough to move hydrogen ions to the outer matrix, but not so much to explode the cell. And that's what happens in our series of reduction, oxidation, reactions, or redox. And here's what those guys look like. So this membrane here is the crystine. Right? This would be the inner matrix where we did our citric acid cycle. And this is the outer matrix with the outer membrane of the mitochondria up there. So this first molecule is our NADH. It's come in with the hydrogen. The FAD, although it's at a lower energy level, has a greater attraction for the hydrogen. Okay? It's at a sufficient lower energy level that there's enough energy in there that we can actually pump hydrogen ions when we split the hydrogen to a proton and an electron, we can pump those hydrogen ions to the outer matrix. After the hydrogen leaves the FAD, it's split into the electron and the hydrogen. And now we go through a series of molecules known as cytochromes. They have pigments in them, but they also have an attraction for the negative charge of the electron. And as we go through a series of them, back to that previous diagram that I drew on the board, I did draw the board, but these components. So the electron is accepted and then given off, and then accepted and then given off, and then accepted and then given off. All right? As we go through various lower energy levels. And that's what we mean by reduction-oxidation reaction. Now, I know you guys know the oil rig kind of terminology. I actually remember it a slightly different way. You guys heard of oil rig? So, oxidation is a loss of electrons, and reduction is a gain of electrons. The way I actually remember it is, let's say that I have cytochrome B, and now an electron is going to be removed, and that is going to give me 3 positive charges. Okay. And let's say I go to cytochrome, we'll choose A, so one that's not the next one in the cycle. But now I add an electron, and I reduce the number of positive charges. That's how I always remembered it. Okay? But this is probably easier for you guys. I'm just stuck in my ways. And so in this process here, where we go from here to here as we remove an electron, so this is a loss of an electron, it's becoming oxidized. Okay? Now if it helps, does oxygen want electrons or does it want to give off electrons?
It wants them.
It's stabilized by the addition of electrons. So if you think of oxygen wanting electrons, it's pulling electrons away from other molecules, so that's how you can think of the term oxidized. The oxygen is taken away the electron. Now obviously, it's not always oxygen in this sequence, but whatever helps you kind of remember what's going on. And so the next molecule, so we had cytochrome B, cytochrome C sub 1, cytochrome C, cytochrome A sub 3. So when the electron is added to the cytochrome B, it is reduced. And then the cytochrome C1 removes it, and it is oxidized while the cytochrome C1 is reduced. Okay? So I do want you to be able to recognize, if I give you this sequence, and I say that we're taking this electron and bringing it here, say to cytochrome C, then is this molecule being oxidized or is this molecule being reduced? So this one would be oxidized while this one is reduced. So this one was this, and now it's being reduced to that, and that one's being oxidized to that form. This would be the oxidized form. This would be the reduced form. And again, the reason why that occurs is because of the greater affinity for that electron. All right. So that's the reduction oxidation component of what's going on here. And this can be a little overwhelming when you look at these pictures and you don't know what the letters mean and so on. But this is the NAD, this is the FAD, and then these are the various cytochromes. Some of them function as transport They kind of carry the electron to the next major molecule. So these three major molecules here, ubiquinone and our NAD, are the sites that actually function as pumps using the energy, kind of like we have a water storage tank, and we pump the water up to the top of the water storage tank. So that the energy that's being released as we move into lower steps is being used to move the hydrogen to the outer matrix. All right, we have enough energy at these three different locations to do that. Now, what happens if we let all that water out at once from our water storage tank? It's going to have a great force, okay? And we can capture this force. That's what hydroelectric plants are all about. As the water comes over a waterfall and spins turbines. And then those, I'm not a physicist, but there's magnetic coils and so on that are used to generate electricity. So this molecule right here is known as ATP synthase. And there's videos that are animated you can watch online that as the hydrogen ions flow down through there actually cause this molecule to spin. And the energy that's that's created by that flow of charged particles is used to bind a phosphate to an ADP. The third phosphate is our high energy area, and that force is used to create the ATP molecule. Well, we've got this hydrogen in on this back inside the matrix again, and we've got our electron now at its lowest level. All right, that's the guy living on the beach. And, well, actually, girl, the guy living on the beach is our oxygen. And those are combined together, the hydrogen ion and the oxygen, receiving that to form an end product of water. So, as we go back through and look at glycolysis, what would be end products of glycolysis?
Pyruvate.
The pyruvate.
NAD.
The NAD with the hydrogens.
And
And ATP. Okay. When we look at our citric acid cycle, what are the end products of our citric acid cycle?
CO2.
CO2,
FADH2.
FAD and NADH2,
GTP.
GTP, which is equivalent of ATP, and the oxaloacetic acid or oxaloacetate. Because we have to end up with our starting point again. Okay? With electron transport, what's our end product?
ATP and water.
ATP and water. Okay? Obviously, along here, some of that energy is not captured and is heat. Because in this process, about 50% of the energy that's being released and all of this is not captured in the formation of ATP and remains as the energy in the energy form of heat. Okay. So, what would be our final products of the entire glycolysis, citric acid cycle, electron transport?
ATP and water.
ATP, water, CO2, and heat. Okay. So oxygen is not a product. Oxygen is coming in with the water. It's forming the water. Okay. So this can get very complicated. When I was in graduate school for my master's, one of my roommates was a medical student, and our entire shower was formed of this kind of stuff and plastered all over the inside bathroom door and forming wallpaper were all these biochemical maps, not just of this, but a lot of it was, with all the enzymes and the molecular. So my son in high school had to learn the molecular structures of all the different steps of glucose and everything else. So we can make it as complicated as we want, but I want you to see the purpose of it and just the major steps. So again, focusing on the summaries that are listed for each of those. Okay? Now, here's a view of that spin as the hydrogens come through. This is a view of the energy level, but I'm moving on here to, okay. So some terms. After glycolysis, anybody know what glycolysis means?
Glucose lipase.
Glyco is referring to glucose, although it sounds like glycogen, sugar. And lysis is
Break apart.
breaking apart. So it's the breaking apart of sugar, right? Which is what we do in most cells, except for the liver. So as we absorb simple sugars across the columnar wall of the small intestine, our blood levels of glucose are going to rise. And as we'll talk about on Tuesday, that's going to raise levels of the hormone insulin. And the liver is one of the primary cells that lowers those levels of glucose and stores the glucose as glycogen, right? So it goes through this process where it phosphorylates that 6 carbon. That is an irreversible process in the cell. So when we were talking about glycolysis, and we added that first phosphate group to get to glucose 1-phosphate, glucose 6-phosphate. That's not reversible, okay? But it is reversible, so the cells are committed now to completing that energy step with the glucose molecule. However, in the liver, it doesn't want to process all the glucose and make ATP. The cells have to do that for themselves. And so that first step is reversible in the liver. And then we go through further steps to form the storage back to starches, if you will. Glycogen is a polysaccharide, right? So we have these terms, glycogenesis and glycogenolysis, to add. So glycolysis is the anaerobic first step in processing glucose to make ATP. In the liver, we do glycogenesis, or rather, and this is because we're looking at glycogen, not glucose. So glycogenesis is the formation of polysaccharides, glycogen, from stored glucose. The purpose of this being that if you don't eat for another 3 to 5 hours, you have this source of glucose that your body can release. And so we have that process of release is glycogenolysis. So this is the breakdown or catabolism of glycogen back to glucose. Which is why that phosphorylation step needs to be reversible. Now we do have the ability of utilizing other molecules, such as lipids and amino acids, to pass through our glucose cycle. All right? So we have another term, gluconeogenesis. So this is the utilization of alternate fuel sources for either glycolysis or the citric acid cycle. So if we look at lipids, so yesterday when we were using pancreatin, it was the pancreatic lipase specifically, and there that we were utilizing to break triglycerides down into monoglycerides and fatty acids. So the glycerol, being a 3-carbon molecule, can come into the glycolysis, and by adding a phosphate group to it, form glyceraldehyde 3-phosphate. And the body doesn't know the difference from that point on, and it can continue through pyruvic acid to the citric acid cycle. Fatty acids, remember, are long chains of carbons with hydrogen. And they undergo a process, enzymatic breakdown, forming 2 carbon molecules. Our acetyl groups, after the intermediate step, are 2 carbon molecules. And so this is known as beta oxidation. So beta oxidation can come in and introduce those 2 carbon acetyl groups to the citric acid cycle. So individuals who are diabetic and are unable to take up the insulin and take up the glucose sometimes resort to breakdown of lipids and increased fatty acid, increased beta oxidation can cause them to go into ketoacidosis. They often, when they're in ketoacidosis, act like they're drunk. I was in the emergency room once with my oldest as an infant. He couldn't walk, and I needed to water some trees, so I plopped him in the middle of the grass thinking he'd be safe while he went around and watered the trees. And I came back, and he had a partial mushroom in his hand and the rest of it in his mouth. I was pretty sure it wasn't poisonous, but wanted to make sure, so I took him into the ER where they flushed out his stomach and charged me $50 for a syringe that they had the charcoal in. And I had to sit there for an hour while they waited to see what was going on. They charged $400 for that, just to sit there and wait. That was 20-some years ago. But anyway, while I was waiting, a woman came in under suspicion of drunk driving, and when they did blood tests on her, she was actually in ketoacidosis. And these individuals often have a fruity smell. I don't know if any of our tin urine smelled acetone, fruity. We have a hard time getting some of that chemical to add to the urine. But that's the release of additional, as someone who's had to go through fatty acid breakdown and beta oxidation a lot. Now, for those people that are trying to lose weight, this is a good way, it seems, to do it, is to break down fats instead of sugars. So on Tuesday, when we look at growth hormones, well, one of the first ones we're going to look at is growth hormone. And it seems to be the ideal weight loss kind of supplement because it takes amino acids from the blood, uses them for energy as well as building proteins. It switches from using glucose to using fat, so you have fatty acid breakdown. So you're building muscle when you're getting slim. What more could you ask for? And one of the case studies, I don't think I put it up on our list, one of the case studies has to do with about 19 or 20-year-old bodybuilder who was taking growth hormone supplements and was doing well, building the muscle, losing the weight. But after a couple of years of doing this, he became a diabetic. Because what it does is it switches the body from using the glucose to using the fatty acids. The glucose builds up in the blood, and you basically wear out the pancreas, producing additional insulin, trying to lower levels of blood glucose. So that would be a form of diabetes type 2, diabetes mellitus type 2, rather than the autoimmune component, where the pancreas is just stressed out, trying to produce increasing amounts of insulin.
Beta oxidation just adding.
Beta oxidation is the specific breakdown of the bond from every other carbon. So you end up with 2 carbons. Obviously the hydrogens are on there as well, but you end up with these acetyl groups. I thought I had a slide in here for proteins. Amino acids. This is still just lipids. Amino acids typically come in through the citric acid cycle as they're modified. This is a slide just kind of summarizing all of those together. All right? So that's kind of what I want you to know about this, is where in our glucose breakdown could we bring in other foods. So we really don't want to use amino acids for fuel that in the body ends up robbing itself. But this is what happens in cortisol. We have high levels of cortisol and adrenal tumor. We'll talk about on Tuesday. Amino acids are broken down. Individuals get weak in their extremities. And amino acids are used for fuel as well. Now, a couple of terms that I want to introduce you here to you are, and I'm sure you've heard these if you took nutrition, but we have the absorptive state and we have the post-absorptive state. All right, absorptive refers to what you're doing after you eat a meal. So the absorptive state, depending on how much lipid is in your meal, is going to be up to the first 2 hours or so. It could be longer if you have a lot of adipose tissue that you're eating. And then we have the post-absorptive state. So what we would expect in the absorptive state as far as blood levels is we would have high levels of glucose, high levels of amino acids, and lipids. And then in the post-absorptive state, we reach a low. Right? So when we talk about hormones on Tuesday, when we talk about the pancreas, what would be the hormone from the pancreas that you would associate with the absorptive state?
Pancreas.
Enzyme. I mean the hormone, not enzyme.
Insulin?
Insulin. So with high levels of glucose, we would expect to have insulin active in the absorptive state. So sympathetic or parasympathetic nerve as well being involved here.
Parasympathetic.
Parasympathetic. So you have the vagus nerve is going to be responsible for release of insulin, as well as the simple presence of glucose in the blood will trigger that. All right. Then when we reach the post-absorptive state, where we have low levels of glucose, then glucagon is going to be responsible also from the pancreas for triggering the glycogenolysis and release of glucose from the liver. And so rather than being parasympathetic, now we are not doing the normal digestive. These would be sympathetic nerves that are working on the liver. Okay, so we have both hormonal and neural control over that. Let's finish with this page. And identifying, obviously there's a whole ton more nutrition stuff to go into, but so a calorie versus a kilocalorie. What's our definition of a calorie? We're talking about energy levels here. And this is calorie spelled with a little c.
Isn't the amount of energy required to...
That's a calorie with a
Yeah.
C.
Amount of energy measured in BTUs, which is British thermal unit, required to raise the temperature of how much water?
1 gram?
1 mil, well, it should be 1 gram because it's distilled water, and 1 mil of water,
Probably 1 degree.
1 degree Celsius.